Hello everyone and welcome to Crack Grade B. In this video, we will be studying about pasteurization. So here are the subtopics that we are going to cover under pasteurization. Introduction to pasteurization, difference between pasteurization and sterilization, about D value, Z value, F value and TDT, types of pasteurization and advantages and disadvantages of pasteurization. So let's start with introduction to pasteurization. What is pasteurization? Pasteurization is a food processing method where a mild heat treatment is applied to foods. For example, milk, fruit juices and some beverages that destroys or kills pathogenic and harmful microorganisms and extends the shelf life of the food. In simple words, pasteurization is a preservation method where a mild heat treatment is given to the foods. Definition by Codex Alimentarius Committee for Food Hygiene. Pasteurization of milk is defined by the Codex Alimentarius Committee for Food Hygiene as microbiocidal heat treatment aimed at reducing the number of any pathogenic microorganism in milk and liquid milk products if present to a level at which they do not constitute a significant health hazard. Pasteurization conditions are designed to effectively destroy the microorganisms Mycobacterium tuberculosis and Coxella burnetii. Pasteurization conditions are designed to effectively destroy the microorganisms Mycobacterium tuberculosis and Coxella burnetii to protect the health of the milk consumers. Previously, the index organism of pasteurization was Mycobacterium tuberculosis as it was considered most heat resistant, but later Coxella burnetii was found as the most heat resistant organism of public health significance. Now, Coxella burnetii is considered as the index organism for pasteurization. Epidemiological studies has interpreted Coxella burnetii as a significant pathogen causing clinical diseases through the ingestion of milk. It is a foodborne pathogen. Mycobacterium tuberculosis causes tuberculosis and Coxella burnetii causes Q fever. The process of pasteurization was named after French microbiologist Louis Pasteur. Louis Pasteur found that heating some liquids such as wines to a high temperature improves the keeping quality, and hence the wines can be aged without sacrificing their fine quality. Pasteurization of milk came into use in commercial scale in late 1800s. Pasteurization is mainly used in dairy industry, especially in milk pasteurization. The other products which can be pasteurized are fruit juices, non-carbonated beverages, beer, liquid eggs, etc. Adequacy of pasteurization in milk is checked by the alkaline phosphatase test. Alkaline phosphatase is an enzyme that is naturally present in raw milk, which can be used as an indicator of proper milk pasteurization. Complete pasteurization will inactivate the enzyme below levels which are not detectable by conventional method. The phosphatase test is used to detect inadequate pasteurization. Sterilization. Sterilization refers to the process which removes, kills or deactivates all the forms of life, particularly referring to the microorganisms such as fungi, bacteria, viruses and spores also. After sterilization, an object is referred to as being sterile or aseptic. Aseptic means it is free from contamination caused by harmful bacteria or other microorganisms. Surgically sterile or sterilized. Differences between pasteurization and sterilization. Pasteurization eliminates pathogenic microorganisms, whereas sterilization eliminates all the forms of microorganism. Pasteurized products have shorter shelf life, whereas sterilized products have longer shelf life. Pasteurization is mostly applied in food industries and sometimes in treating medicines, which may include some thermolabile ingredients, whereas sterilization is used in making the instruments of the surgery sterile. It is also applied in microbiological labs for treating the culture mediums and also used in food industry, packaging industry, etc. Decimal reduction time, D value. The D value denotes the decimal reduction time. It is the time required at a specific temperature and under specific conditions to reduce the microbial population by 90% or by 1 decimal, or we can say by 1 log cycle. This is a graph from which we can calculate the D value. The y-axis shows the number of survivors, while the x-axis shows the heating time in minutes. When a medium is being heated for 7 minutes, let the load of an hypothetical microorganism be 10 to the power 4. After 13 minutes of heating, for example, the microbial load reduced to 10 to the power 3. So for this 90% reduction, it took 6 minutes. So we can say that the decimal reduction time was 6 minutes. And this whole condition was done under a specific temperature, or we can say a constant temperature. The D value depends on the temperature, the type of microorganism, and the composition of the medium containing the microorganism. Thus, after an organism is reduced by 1D, only 10% of the original organism remains in the medium. So, 10 to the power 3 by 10 to the power 4 will show that it is the 10%. Okay, so 10 to the power 3 is the 10% of 10 to the power 4. So we can say that only 10% of the microorganism is present in the medium. When referring to D value, it is proper to give the temperature as a subscript to the D, because D value for different temperatures will be different. For example, a hypothetical organism is reduced by 90% after exposure to temperatures of 300 degree Fahrenheit for 2 minutes. Thus, the D value would be written as D subscript 300 degree Fahrenheit is equals to 2 minutes. Canned foods are susceptible to the spores of the organism Clostridium botulinum. This is the organism that causes botulism. So, as the spores of the Clostridium botulinum are highly heat resistant, it requires a 12D reduction or 12 log reduction at 250 degree Fahrenheit. Z value. The Z value is the increase or decrease in temperature required to reduce or increase the decimal reduction time by 1 decimal. In other words, the Z value of an organism is the temperature in degree Fahrenheit that is required for the thermal destruction curve to move 1 log cycle. This is the graph which shows the temperature D value, and these are the number of survivals, and this part shows the Z value. So, if we increase the temperature, for example, from 235 degree Fahrenheit to 255 degree Fahrenheit, then the decimal reduction time will decrease. In simple words, if we increase the temperature, then the time required for the destruction will decrease. That is why it is said that Z value is the increase or decrease in temperature required to reduce or increase the decimal reduction time by 1 decimal. This means when we need to reduce the decimal reduction time, we need to increase the temperature. And when we need to increase the decimal reduction time, we need to decrease the temperature. F value. F value for a process is the number of minutes required to kill a known population of microorganisms in a given food under specified conditions. Note, the actual processing time a can of food is given in a retort is always greater than the F value due to the heat penetration requirements. Thermal dead time, TDT. Thermal dead time is the amount of time that is necessary to kill a specific number of microbes at a specific temperature. The value is obtained by making the temperature constant and measuring the time required or necessary to kill the specified number of cells. Types of pasteurization. There are 3 types of pasteurization that we generally use. Low temperature long time, LTLT. Temperature requirement is 63 degrees Celsius or more. Time is 30 minutes. High temperature short time, HTST. Temperature requirement is 72 degrees Celsius or a bit more. Time 15 seconds minimum. At some places they follow 15 seconds as minimum time, but we generally follow 15 seconds as per FSSAI. Ultra high temperature UHT, temperature requirement is 135 degrees Celsius and time is 1 to 2 seconds. In bottle pasteurization. Bottles are filled with raw milk and then they are tightly sealed with special caps and are kept at 63 to 66 degrees Celsius for 30 minutes. Then those bottles are passed through sprays of decreasing temperature which cools the bottle and the product. Advantage. Post-pasteurization contamination possibilities are less. Disadvantage. Transfers heat very slowly. Mostly glass bottles are used, so chances of breakage are high. Batch or holding pasteurization. This mainly refers to the low temperature, long time LTLT method. The milk is heated at 63 degrees Celsius for 30 minutes and then cooled to 4 degrees Celsius or below in this system. There are 3 types of batch pasteurizer. First one is water jacketed vat. This is double walled around the sides of the vat through which hot water or steam is passed for heating and cold water for cooling. Next one is water spray type. A film of water is sprayed from a perforated pipe over the tank holding the product. Third one is the coil vat type. The coil containing the heating or the cooling medium is revolved when the product is placed inside the vat. High temperature short time. This type of pasteurizer has many important parts such as a float control balance tank FCBT, pump, regeneration section. Regeneration section are of 2 types, regeneration heating and regeneration cooling, plates, filter, flow diversion valve and etc. Generally, plate heat exchangers are used for the pasteurization of the products. Flash pasteurization is also called the high temperature short time pasteurization. Advantages. Capacity to heat treat milk quickly and adequately, requires less floor space, easy to clean using CIP system. CIP is clean in place system. Disadvantages. This system is not well adapted to handle small amount of products. Ultra high temperature pasteurization. This type of pasteurization has a temperature time combination of 135 degrees Celsius or a bit more for 2 seconds. It is also referred as no hold process. The UHT treatment is successful on immediate aseptic packaging. Some other types of pasteurization are vacuum pasteurization, ultra-pasteurization, statsanization. Last part, advantages and disadvantages of pasteurization. Advantages: harmful pathogens are destroyed, it inactivates the enzymes, it increases the shelf life, decreases the overall microbial load. Disadvantages: cannot destroy spores, it reduces the cream line or cream volume, it fails to destroy bacterial toxins, it makes difficult to clot the milk using rennet, it may be used to mask low quality milk. Thank you and happy learning.